presentation talk to people that are interviewing with us, as all of our residents who've been here know, is that you need to try to find a place for your residency that fits you. And this is true with surgery, but it's also true for pediatrics and family medicine or whatever else that you might be interested in doing. And Dave was extremely well thought of by our faculty here. We very much wanted him to do his residency, but he sat down with me at least and said, look, you know, I want to tell you I really like it here, but I do have an interest in doing some basic research. His father is a researcher. And he said, I really think that I want to try to find a program where I can probably spend a couple of years in the lab doing some research. And he said, and besides that, I'm about to marry the number one student in our class, and she is dictating where we're going to go to do our residency anyway, so I really don't have a lot of choice in that. Now, how true that is, I don't know, but that's what he told me at the time. And so he did go off to Duke and did his residency, and during that time he did a lot of research, and I actually happened to see a good bit of it presented because every year at the Southeastern Surgical, he wouldn't even have a faculty member. He'd show up and present a paper at Southeastern Surgical on some topic. So he did incorporate becoming a surgeon along with his research activities. But he told me when he was there as a student, he said, but I do want you to know that depending on how things turn out, this might be a place I'd like to live someday. And so he ended up at Vanderbilt then doing his pediatric surgery training after residency, and sure enough showed up and said, okay, I think I'd like to come live here, and we were successful in recruiting him here. So he was a good example of somebody who figured out what the fit was going to be and did it and took full advantage of it, and we're just proud to have him back here. And I know our residents are proud because on research he helps Dr. Stanley a lot in terms of the research activities. So Dave, glad to have a chance to introduce that to the students and look forward to this talk.
So good morning. Thank you, Dr. Burns, for that introduction. So this is it's going to be a speed pass through what I did for about 5 years in my general surgery residency. So the way they had it for us is that after your intern year, you start writing grants to get funding to do your research. So your second clinical year, you're running through the ICU and taking consults and writing grants at night. And then your third and fourth year, you're in the lab. And then fifth and sixth year, you're cleaning things up. And seventh year, you're applying for fellowship. So this is kind of a 40-minute talk on 5 years of research. So in brief, as you see, the topic is osteopontin regulation of epithelial to mesenchymal transition and tumor metastasis. And I'll explain all those terms to you. Tumor metastatic progression is where cells, which are otherwise quiescent, undergo this modified polarity so that they can migrate through the basement membrane of the tumor where they're residing and be able to mobilize to a secondary site and incorporate into a secondary place, forming metastasis. This transition from an epithelial cell to a mesenchymal cell has more stem cell-like properties, is called EMT, or epithelial to mesenchymal transition. Several of the widely metastatic tumor types, such as metastatic HCC, renal cell, breast cancer, and pancreatic cancer, the most metastatic subtypes of those tumors all have very high pro-EMT properties, and those are noted by cell surface receptors for TGF-beta, Wnt, Notch, Hedgehog. And these are all independent pathways to perform epithelial to mesenchymal transition and have implications for tumor metastatic phenotypes. So EMT, how do you know if a cell is undergoing EMT? It has marked E-cadherin expression, and they also desolute their ability to adhere to one another. So you can imagine that tumors kind of grow in a sheet, and a cell that wants to migrate to a secondary site kind of has to detach from its neighbor, get into the bloodstream, go somewhere else, find a soil. It's called the seed in the soil theory, but find some soil for that seed to grow and grow a secondary tumor site. So that process, you can note that a cell is about to do this by their expression of E-cadherin, as well as other mesenchymal markers, vimentin, N-cadherin, and fibronectin. So in these stem-like characteristics that you can tell on just gross morphology. They become more spindle-shaped as opposed to a columnar cell, as well as their resistance to apoptosis. And these are all not only critical for tumor progression and metastasis, these are physiologic roles. This is how you develop as a human embryo. So part of developing into a human embryo, your hand develops as a pad, but the reason that you have fingers is because EMT allows the digital webs to form. So these are all normal processes that somehow are overtaken by a metastatic pathway. So osteopontin has been noted to be, it has physiologic roles as well, and we'll talk about that, but it was noted in the early 80s, for some reason, women with highly metastatic phenotypes of breast cancer have very high serum levels of osteopontin, and nobody had really made the link as to what osteopontin was doing, but a woman with triple negative breast cancer with metastatic mets or brain mets, their serum levels of osteopontin were very high, and those tumors had more osteopontin production at the periphery of those tumors. So osteopontin is a secreted phosphoprotein with physiologic roles for bone remodeling, hence osteopontin, immune modulation, embryogenesis through resistance to apoptosis, as well as cellular differentiation. And we know that it functions by binding cytokine receptor CD44, and there's another receptor for alpha V beta 3 integrin. And this introduces an intracellular cascade of signal transduction mediation. So kind of osteopontin has two different binding sites, and this will be important for the rest of the talk. So increased osteopontin expression, kind of, you'll go back to remembering what I said earlier. Osteopontin itself increases a tumor or a cell's ability to invade and migrate. It increases the cell's ability to avoid apoptosis, improves angiogenesis in the local tumor stroma. It reduces the tumor's ability, it reduces the tumor's susceptibility to the innate immune tumor response, as well as promoting tumor growth. And you'll note these are all traits that permit metastatic progression of tumors. The other interesting thing, this was noted in the Journal of Cell in 2008, is that osteopontin is not only able to stimulate the soil around the tumor, but it also can act in an endocrine function and go into the bloodstream and stimulate bone marrow to secrete stem cells. So it helps mobilize stem cells to secondary sites for metastatic tumor preparation. So it primes the soil for the seed of the metastatic tumor cell to grow. And what are mesenchymal stem cells? So they're pluripotent progenitor cells that are physiologically used to remodel sites of inflammation, but pathologically they promote remodeling and the priming of the soil for malignant cells. They can either be locally derived, so you have mesenchymal stem cells throughout your body. If you have a tear in your muscle after you've worked out, then those stem cells help knit that muscle back together, but they're also derived from the bone marrow as well. And as we talked about, they promote cancer growth via both paracrine locally and endocrine through the circulation signaling pathways. As I was joining the lab, my mentor, who subsequently went on to be the chair at Loyola and is now the chair at University of South Florida, he had been working on this for about 15 years before I came to the lab, and he had never nailed down the link between cancer cells and stem cells and osteopontin, but he had multiple different people working in his lab, and he tasked one group of scientists to create a drug. It's not really a drug, it's an aptamer, but it can be modified into a drug. And an aptamer is just a group of highly selective binding, and they're non-immune amino acids, so essentially a 23 chain of amino acids which can very avidly bind to the active site of osteopontin. So essentially osteopontin has this receptor which binds to the alpha V beta 3 receptor or the CD44 integrin receptor, but this aptamer R3 fits right in there like a lock and a key, and it prevents it from being able to bind anywhere else. So essentially it renders osteopontin inactive, and that was really the unlinking for all the rest of my research. So as I say here, it is highly selective, binding to osteopontin at the alpha V beta 3 integrin receptor site. So the first thing that we had to do is kind of prove this link between metastatic cancer cell lines, osteopontin, and this pro-metastatic phenotype. So we grew MB231 cancer cells, which is a highly metastatic human cell line of triple negative breast cancer, and we grew them in a petri dish or a cell culture dish. And we were able to show that metastatic phenotype was arrested. The cells behave differently when you could block the osteopontin. These MB231 cells make a lot of osteopontin endogenously. And we proposed that this was through the highly selective binding site of the alpha V beta 3. So it's kind of a busy slide, but if you just grow these cells in and of themselves in a dish, we have adhesion, migration, invasion. These are all surrogates for tumor metastases. And if you just grow them in a dish alone, this is their rates of adhesion, migration, and invasion. When you add even more osteopontin, you really don't change the way that they adhere, migrate, or invade. So adding more osteopontin back to a cell line that already creates osteopontin doesn't really change anything. So it's not really dose dependent. When you add the osteopontin aptamer, so by blocking the osteopontin's function, you see you have significant, those little asterisks denote decreased adhesion, migration, and invasion. And then, kind of being good scientists, we did a host of negative control studies. So adding osteopontin but degrading the osteopontin with RNase, or blocking the CD44, or just blocking the alpha V beta 3 antibody, so just taking that protein out didn't change anything. But you can see by adding the medication or the aptamer to block osteopontin, it decreases those cells' ability to adhere, migrate, and invade. So that's a surrogate for metastatic phenotype. Well, what does this mean in a real human or in a real mouse? So the next thing I did is I gave 6-week-old non-obese SCID mice, I injected 2 million MB231 triple negative breast cancer cells in the mammary fat pad of these mice, and I labeled it with a luminescence tag so that every week I could image these mice. I would anesthetize 60 mice every 3 days and take pictures of them in this luminescence chamber. But you would see that when you don't treat them at all, the breast cancer cells grow avidly as well as metastasize to the liver in this case. When you treat them with a sham aptamer, so it's an aptamer that we know does not work, it looks very similar to the non-treated group. Well, if you take those same mice and you treat them every 48 hours with a tail vein injection, I was the chemotherapy nurse for these mice for 6 weeks, you see their tumors don't grow very well and they don't metastasize. So this is just a graphical representation of the tumor growth as well as the tumor volumetrics in the untreated and the sham treatment group as well as our treatment group. So this had not only implications in the petri dish, but it also had implications in a real animal model of human breast cancer cell lines. So the next thing, also kind of at the same time as that Cell paper came out, there's a paper in Nature looking at the way tumor cells interact with mesenchymal stem cells. So they showed in this paper that breast cancer xenografts, even if you have a weakly metastatic phenotype, so a triple positive breast cancer cell line, if you just inoculate those with stem cells at the same time, you take an otherwise quiescent cell line, you create it into a very avidly aggressive breast cancer cell line. And they didn't make the link between osteopontin, but they made a separate link. They said that this happens because the cancer cells interact with the stem cells, and this is through CCL5 or chemokine ligand 5, and that increases mobility, invasion, and metastasis. And when I read this, I said, well, this is all the same language that I'm using for my cell lines, so why is there some link back to osteopontin that's creating this function? So, and they noted that MB231, which is the same cell line I'd used previously, when you co-culture them in the same petri dish with mesenchymal stem cells, they increase their CCL5 expression. So what we wanted to do is see if osteopontin somehow mediates this crosstalk between tumor cells and mesenchymal stem cells. So we know that tumor cells talk to stem cells, and stem cells increase CCL5, and that increase in CCL5 creates these stem cells to change into cancer-associated fibroblasts, which create kind of the soil for tumors to grow. So can we prove that this is osteopontin mediated? And then can our aptamer abrogate or stop that crosstalk and essentially block the conversation between the stem cells and the cancer cells and prevent that mesenchymal stem cell from changing into a cancer-associated fibroblast? and we'll go through more of this. So the first thing that we did was we just cultured stem cells in a dish and just added osteopontin. We didn't add any other cells. We just said, hey, can osteopontin in and of itself stimulate these mesenchymal stem cells to create CCL5? So that's what we did. We found the serum concentration of osteopontin in metastatic breast cancer patients, and we dosed these stem cells with that same concentration. We also had positive and negative controls, so we dosed these stem cells with osteopontin, and we blocked that osteopontin with the aptamer, and we also had sham groups. And then after 72 hours of growing these cells, we would kill the cells, extract their protein, and look for markers for cancer-associated fibroblast changes, which is CCL5, as we talked about, and then other things that we thought could be the cause, which was MCP1, IL-6, and MIP. And I won't go through the background, but those are other things that were thought to maybe cause this change. Well, as you can see here, so if you just grow mesenchymal stem cells in a cell chamber and you do nothing to them, they make a baseline level of CCL5. But when you stimulate them with osteopontin, it nearly doubles their CCL5 expression. When you add osteopontin, but you treat them with the aptamer, it also decreases their CCL5 expression. And we saw no other changes in the other proteins that we thought could be causing this. So again, we locked in that osteopontin seemed to stimulate CCL5 expression, and we seemed to be able to block this expression and this change into cancer-associated fibroblasts by adding our aptamer. And then looking at the other possible pathways, again, we found that by blocking the alpha V beta 3 receptor with and without our aptamer also blocked the CCL5 expression. So these are stem cells alone. You add osteopontin, it goes crazy. You block the osteopontin with aptamer, which we kind of changed routine calling it R3 and RGD, and then treating it with the aptamer as well. And then our negative controls didn't have any change. And then, so not only do you look at proteins, you look at the functional assays as well. So when you're going to try and publish a paper in a high-level journal, you don't just say, oh, this is some vague association. You have to say, I found it in the cell line, I found it in the cell line after I treated it, I found it in the cell line after I treated it, and it didn't change with negative controls. I found it in the cell line, I treated it, I gave it to a mouse, I killed a bunch of mice. So you can't, nature's not going to take the paper where you just show this association. You have to have 17 different layers to prove your hypothesis. So if you find that I'm iterating myself, it's because you have to prove this along multiple different variations. So we did functional assays, these mesenchymal stem cells, to see if they behave like cancer-associated fibroblasts. So stem cells alone don't really adhere, migrate, or invade very well. When you add osteopontin, it kind of goes wild. You can block this function by blocking RGD, aptamer, and CCL5, but all the other sham controls do not have that same expression. And this is a whole nother nerd thing that I went off onto, but essentially we showed that the way osteopontin promotes CCL5 expression in mesenchymal stem cells is by binding to RNA polymerase II. And then nobody had ever proven what that RNA polymerase II was. So using a chromatin immune precipitation assay, we found that it was an AP1 C-Jun homodimer, which nobody really cares about. But I was growing mice all day long, and I had about 3 months to kill while I was being the anesthetist and chemotherapy nurse. So this is something that we did on the side. So going back to our tumor model, again, we created xenografts, or gave mice triple-negative breast cancer cell lines in the mammary fat pads and treated them like I showed you previously, and showed this tumor growth through bioluminescence. Well, the next step was to prove that, okay, let's give these triple-negative breast cancer cell lines and co-inoculate them with mesenchymal stem cells. And this is something also that had never been done. I labeled these mesenchymal stem cells with GFP, which is a green fluorescence protein. So not only could I image these mice every couple days for the tumor burden, but I could also see, kind of using a mini mouse CAT scanner, where these stem cells went. So I know I injected them in the mammary fat pad, but I could track these mesenchymal stem cells and see where they traveled to. So that was kind of a novel technique. And actually, I wrote a paper just about the technique of labeling mesenchymal stem cells for recovery later on, and you'll see why that's important. But I dosed mesenchymal stem cells with these triple-negative breast cancer cell lines, and it's kind of, if you take this all in one shot, it's kind of a lot, but just look at the control. So breast cancer cells plus mesenchymal stem cells, how avidly they grow and how widely they metastasize to the liver and the lung. And let's just look at the breast cancer cells alone. So these are just our two control arms, breast cancer alone and breast cancer with mesenchymal stem cells. And look at these mice. So this mice, the tumor grows definitely, but it doesn't nearly grow as wildly as when you dose them with the mesenchymal stem cells. When you give them the negative control, you can see you really haven't changed anything at all in these two lines. When you treat them with our aptamer, so we've given triple negative breast cancer cell lines with stem cells, and essentially you've been able to arrest this ability of these tumor cells to metastasize, and that again is proven in just the breast cancer cell lines alone. And so when we gave these mice that had mesenchymal stem cells and we treated them with the aptamer, we could actually make them behave like they had never been treated by the aptamer just by giving them back CCL5, which is the whole downstream effect of osteopontin. So osteopontin functions by stimulating CCL5 of stem cells. So even if you block the osteopontin but you give back the downstream promoter, you have a very widely metastatic cell line. So essentially we were able to show that osteopontin not only stimulates mesenchymal stem cells to promote a pro-metastatic phenotype, but we proved that it was through a CCL5 mediation. And this is all graphical representation of those same photographs that I showed you previously. Why this was kind of novel that we were able to label these mesenchymal stem cells is that I did necropsy on all these mice and pulled out the primary tumor, the livers, and their lungs where they had metastases. And the luciferin, or this picture to the left, is the tumor itself. And this picture to the right is you're only looking at the stem cells. So you're looking at the stem cells in the primary site. And then in the liver, you can see these kind of faint, I don't know if it picks up here. This whole time I didn't realize that what I was pointing at on my screen was not showing up to you guys. So the green is GFP at the primary sites, and then you can see here at the liver and the lung, you can see that you have stem cells that have migrated from the primary site to these distant sites. You don't see that in the aptamer treated group, but when you give a sham aptamer, you see these stem cells still migrate to a secondary site. And when you add CCL5 back, you still have them migrating as well. So, and then when you just dose mesenchymal stem cells, they actually kind of never go anywhere. They stay in their primary tumor site. They don't go to the liver or the lung. So what I was able to do was emulsify those solid organs and be able to do a FACS sorting so that you put them through a cell sorter that sorts any cell that's putting off this luciferin light, and we're able to count back the cells that we had. So in the lung, you see for the mesenchymal stem cells that were not treated, you see that they've migrated, but when you treat them with aptamer, the cells don't migrate to the liver or the lung. And again, with the sham aptamer, and then by giving back the CCL5, you see those stem cells migrate back to the secondary sites again. So using those stem cells I was able to recover, then I was able to do quantifiable PCR on those stem cells that had migrated to a secondary site and assay them for cancer-associated fibroblasts. So seeing not only did they migrate, did they really turn into these cancer-associated fibroblasts which prime the soil for the secondary site. So those markers are alpha SMA, tenascin C, SDF1, and FSP. And we do show that they have significantly increased expression of those markers, which prove that they have converted from a mesenchymal stem cell to a cancer-associated fibroblast. So all that to say, kind of a summary of the salient points for my research was that osteopontin interacts with mesenchymal cell, surface integrin receptors to upregulate CCL5 expression. This is done through an AP1 C-Jun homodimer activation of the CCL5 promoter. Co-culturing osteopontin expressing MB231 cells with mesenchymal stem cells induces CCL5 expression and is ablated by our osteopontin aptamer. Xenografts showing co-inoculation significantly increases aggressive tumor growth and metastasis CCL5 levels and migration of mesenchymal stem cells to distant sites. And this was able to be abolished by aptamer administration and restored by CCL5 administration. And we also showed that mesenchymal stem cells recovered from metastases show osteopontin and CCL5 dependent cancer-associated fibroblast marker. And this osteopontin may potentiate tumor growth by interacting with mesenchymal stem cells, increasing CCL5 and cancer-associated fibroblast. So then, not only do you have to prove it in one cell line, so we've done all this work with breast cancer cell lines, then you have to start all the way back at the beginning. And if you're going to make a broad statement saying this works on all epithelial cancer cell lines, I had to do the whole thing over again using HCC cell lines. So I won't bore you with this, but again, doing the same project using HCC cell lines, I did direct injection, did a laparotomy in all these mice, injected their livers with highly aggressive HCC cells and watched them grow for 10 weeks. But you see the control arm here, over 7-week time, you see how wide that tumor grows. When you treat them with aptamer, that tumor growth is pretty stable. And when you treat them with the sham aptamer, you have, again, a widely metastatic and widely aggressive liver cancer cell line. So in summary, this is a novel pathway. This had never been described before, and there's actually ongoing research, and that drug is now in phase 1 clinical trials because it's actually very stable and it can only be dosed once a week. We've methylated it and added all these different sterols to it to allow it to stay in the circulating system of humans for up to a week at a time. So essentially, this is a novel pathway for osteopontin-dependent tumor metastases that was able to be blocked by a drug that was made in our lab, this osteopontin aptamer. So that was kind of a long way of going around a scientific talk, but I found that to be as obsessively interesting to me as surgery was, and I was really glad to have done it. I met so many people. I was able to travel the world giving talks on this on somebody else's dime. So if this is interesting to you, it's a really great way to spend a few years. Ultimately, one could say, well, you did all this research and you're in Chattanooga and you're not furthering on your work. Well, I still talk to my collaborators. I had actually looked at a job at St. Jude and gave this very same talk and had proposed my future progression concepts for the next 10 years and actually gotten it funded through St. Jude. And I decided this was a better use of my time, is to help children every day instead of worrying about how I was going to kill the next mouse and how I was going to get grant funding for the next 10 years. So actually this project is still going on at St. Jude. The guy who took my job instead is a good friend of mine from Boston Children's, and he's actually continuing to do this work on pediatric tumor cell lines. So even though I'm not doing it, I'm still kind of advising and chatting with them and helping them work through their stuff, because I have a little bit more experience than they do with this work. But our careers are a lifelong educational game, and I learn stuff every day, whether you're in Chattanooga or in Boston or wherever you are. There's a lot to be learned. And personally, when I was looking down the barrel of the next 30 years of my career, I wanted to be able to look and know that I've helped a lot of children, and I thought I would have a better chance of doing that than spending my time in the lab. But I think there's a lot of different ways to have an interesting career, so I just kind of wanted to highlight what that could look like.
We have questions or comments from the audience about this? Yeah, Heath.
Dave, that was awesome, and really highlights, I guess, what you can do when you're in in that environment and able to apply yourself to that. My question would be more about the pharmaceutical side. So how does it go from what you guys did, which was novel, to a pharmaceutical company kind of picking this up and trying to develop a drug? And how does that relationship work with patents and the whole deal?
So all these major institutions, St. Jude specifically, Duke, Harvard, they have pharmaceutical silos within the university itself. So actually the vice chair, one of the vice chairs of the surgery department he's not a surgeon, he's actually a pharma guy. And he works in the department and he creates this conversation. All of this has to get vetted through legal before we can even publish the papers. It's got to go to the pharma silo to get patented, and then you can't even write your paper until they've at least applied for the patent with my nucleotide sequences on there. So it's this whole back. It's not a game, I mean, because the institution is really making a significant amount of money. So even though my mentors left Duke and I've left Duke, and Duke still actually owns all this work. And that's how that works. It was above my pay grade, but I knew it was all going on, so.
Yes, Dr. Greer?
The time that you spent, so this is your first 2 years of how did you do this and your residency so far as year by year, and then you did 2 years of this, and then you did 4 years of surgery?
So you do your intern year and they don't really bother you. And then I will never forget that it was July 7th of my second year. It was like literally a couple days in, I was post-call, and the chairman calls you and saying, it's like getting your phone call from Dr. Burns at 2 o'clock in the afternoon, you've just gotten home. And I remember I didn't have air conditioning in my car, and you don't show up anywhere in that hospital without a tie on. So like I'd just gotten home, and he's like, can you meet with me in 15 minutes? I was like, okay. So literally I run, I drive. It was July in Durham, no air conditioning. I like got the windows down. I show up running into his office, and I'm literally, my blue shirt is just sticking to my back. And he's like, have you thought about your research time? I was like, no, sir, I'm still trying to figure out what I'm trying to do. He's like, okay, well, great. So you're just going to work in my lab, and I've got this project, and this is how it's going to go. So that's how that works. So then if you want to go into how this worked at our institution, so that's July 7th, and there were grant deadlines for December 1st, and I submitted 8 grants by December 1st, which were each 12 to 20 pages. So if you weren't operating, you were at a computer either entering data or writing a grant. So that was your
You were
second
still a
year.
resident, you were still doing call and
Yeah,
in addition to the research?
right.
So you don't stop all the resident work and
Well,
just
so
do the
then
research.
your third and fourth years, you go to a lab every morning, and just like you guys, we took call about once or twice a week. And then you go back, and so essentially you do all of your science for 2 years, and you're trying to write papers as you can. And then the expectation is kind of you've written all your papers over the next 2 years. So again, the senior residents, if they weren't operating, we had these offices in the hospital with computers, and you were just writing papers during that time. So
So you had protected time. You that morning you were away from the operating room. You were designated to be in the lab or doing your
for
research.
2 years, we had dedicated
2
researchers.
years.
Yeah.
And how many years total then?
7 years of residency.
Okay,
So we do 6, they did 7. So,
okay.
but I will say that 2/3 of us, myself included, had my salary supported by the grant. So it's not like Duke was shelling out my resident salary and benefits. It was supported by, for me, an AAS grant. My other buddies got the American College of Surgeons Research Grant. I applied for the American Hepatobiliary. I applied for the SSO. So they expected us to put this broad net out, and if you didn't do it, then they would still support you, but the expectation was that you were going to try at least 7 or 8 different ways to get funding.
This
make
is
that
every
happen.
resident.
Every resident, yeah.
Yeah, that's the point that, again, we made when we started is that's a focus of the institution. At the time Dave was there, I happen to know from my time on the Residency Review Committee, which overlapped somewhat with that, Duke University had 10% of all the NIH funding in America at that one institution. So the focus of the school and the institution was research activity. I just got back from a meeting at Stanford, the Halstead Society, and it's a very similar mindset and culture there. Their basic research presentations to the Halstead Society from faculty there were some of it very similar to this in terms of what the focus of a career is. Not to say they don't do a lot of clinical work, but they do. I think one point though, when you look at institutions, Dave mentioned that 2/3 of his salary was paid from the grants, but 1/3 of it wasn't. And so the institution has to have the commitment to pick up that 1/3 and not be picking at you about, we can't afford to have residents doing this kind of work if they're not taking care of patients. I think it's interesting, though, that you did have some calls scheduled during your research time, right?
Yeah, I mean, so mostly it was in the ICU, covering the ICU at night. So
Yeah. Because I've related this, a lot of residents here have heard this because it's a story that I tell a lot during interview time, but years ago, a resident or a medical student very similar to Dave was interviewing here, actually was from the University of Mississippi, and we were very interested in him, and he ended up deciding to go to Vanderbilt. And so as a result of being there and coming to the state chapter meetings and things like that, I got to see him from time to time, and their program very similar, 7 years, at least at that time, it was 7 years with 2 years in the lab. And I ran into him I think they only did 1 year, but at any rate, he was a third or fourth year resident, and he came to the chapter meeting and presented a paper, basic science paper, and I saw him, said hello to him, and how are you doing? He said, well, I'm not doing so well. And I said, what do you mean you're not doing well? And he said, well, I spent my first 2 years doing basically clinical work, and I really got to feeling fairly comfortable with what I was doing, and then they pulled me out and put me in the lab. And he said, so I've just now started back, so this was in late July, so I've been back now doing clinical work for the last 3 weeks, and I'm a fish out of water and feel like I'm starting over again. Because during his time in the lab, he wasn't allowed to do any kind of call. It was totally protected time, and so had no clinical experience at all, and said, you know, it was just like starting over again, which frankly, if you think about the process of how we all learn, you have to do a lot of it, and especially being a physician and especially being a surgeon. And he had gotten started, I always use the comparison of Michael Jordan and foul shots. He'd gotten started doing the 10,000 foul shots, but it stopped for a period of time and was having to start all over again. He went on, by the way, to become an academic cardiovascular surgeon, but he was really, called me several times just to talk about the transition coming back. So I think it was really good, the point coming around to, it was really good that you had some clinical time mixed in during that time.
And they would use us for all the transplant runs too, so
yeah.
we also, because they knew that we were always available.
So you were at least involved in something. So that's something, again, for people to think about in terms of their careers, is that you get away from what you're doing for very long. It's like an athlete, you have to kind of get back to it. Surgery is the ultimate sport. Dave, this was a great talk, great presentation, and I really appreciate the fact that, I mean, I, along with a lot of people, probably had a little trouble following it all, so it's second nature to you.
Well, I'm sorry, I didn't realize, I thought
No,
I was
I'm
pointing,
not
and I
being-
didn't realize that this was right here, so.
No, not being critical at all. I just,
Here I was
but I
doing
think
all
it's
this.
great to see what you've done with your career and with it, and we're just glad that you decided not to go to St. Jude and come here and spend time with us. So we look forward to a long career here for you. It's going to make a lot of difference in the Chattanooga community and in our program. Thanks a bunch.